Good afternoon, everybody, or good morning, depending on where you are. It is 4:20 where I am right now. Welcome to this session of your Human Growth and Development online course. Today we're going to be taking the embryonic period, which is essentially an extension of last time's topic, which was the third week of development. And during this session, we're going to be looking at the different changes and things that happen during this period in terms of what the three germ layers that we learned about last time are going to develop and how and what they're going to become. So there's going to be a lot of detail to cover with this session, so hopefully we'll get through it without any problems. Okay, so let's get started. So, just a brief introduction. The embryonic period is referred to as the period of organogenesis. And just in terms of what that word means, it means formation of the organ systems. So, basically, during the embryonic period, the three germ layers differentiate into tissues and organs, so that by the time the embryonic period is over, the organ systems have completed their formation. In terms of duration, the embryonic period extends from the third to the eighth week of development. All right, so anything between week 3 and week 8 is considered the embryonic period. Now, before we move forward, there's just an important note that I need to make, and that is just keep in mind that at any stage of development that involves a high rate of cell division, whether it's the initial germinal period or this period, the embryonic period, means that the developing embryo is susceptible to malformations. So basically, if something is happening very quickly, anything can kind of interfere with that process and then just completely mix it up. Okay? And obviously, in order for development to proceed normally, we need the different phases and changes to happen normally as well. And anything that changes them will result in abnormal development. So basically, exposure to dangerous substances, let's say if the mother is on a certain type of medication, or, for lack of a better term, if she has any bad habits or so, or for example, if there's a certain defect in the programming of cell division or the control of the process, then this can cause congenital abnormalities to show up during this period. Okay, so as always, we're just going to do a quick recap of where we left off last time. And remember, we ended with the formation of the three germ layers and formation of the trilaminar germ disc. We said that the appearance of the primitive streak within the epiblast causes a new mass of cells to migrate, and the first thing we had was replacement of the hypoblast with the endoderm, and then a new mass of cells formed the mesoderm, and then we ended up with three layers. Remember, anything in blue is always ectoderm, anything in red is mesoderm, and anything in yellow is endoderm. Okay, so let's look at the ectoderm first, okay? Now, if you guys remember last time, we spoke about how there was a mass of cells that organize themselves within the mesoderm centrally and form something called the notochord. Once the notochord appears, this induces a thickening of the ectoderm. So a certain part of the ectoderm begins to thicken, and we call that thickening the neural plate. Once the neural plate appears, this marks the beginning of something called neurulation, which is the process that will give me the primitive nervous system. And these changes start happening at the beginning of the third week. Okay, so basically think of neurulation as more of the first event of the embryonic period. Okay, so once the neural plate appears under the influence of the notochord, it starts growing, and it grows towards the primitive streak. So basically the primitive streak is closer to the caudal end, and then the neural plate will start growing towards it. Now, as it is growing, its sides, so the sides of the neural plate, will start to elevate. So initially it was a flat structure, and as it increased in size, the lateral parts of the sides of the neural plate started to elevate. These form structures called the neural folds. And with the appearance of the neural folds, we also have a midline depression called the neural groove. Now, what happens is neural folds start or keep elevating, and they grow towards each other until eventually they fuse in the midline. Okay? The fusion happens right in the middle of the length of the embryo, and then it starts to continue craniocaudally, meaning that it continues towards the head and it continues towards the lower body. Okay, so let's just look at that in picture form. We're going to take this in order, obviously, from A to D. Okay, so this is a cross section. Okay, and what they've done is they've left the ectoderm here. This is this layer, and then anything in green is the neural plate. All right. In this first picture, you can see how the sides of the neural plate are starting to elevate, and they formed the neural folds. And this depression in the middle between them is called the neural groove. Okay? Now in the next picture, excuse me, you can see how the neural folds are sort of growing towards each other. Okay? And then eventually they'll get close enough that they actually fuse. And once they fuse, this layer detaches, or this new formed tube detaches. Okay? And now notice how you've got a notochord, and you've got a newly formed tube on top of it. Okay, now this newly formed tube is called the neural tube, and this will end up becoming the nervous system. Now, what you're looking at here is an aerial view. So you're basically looking at the embryonic disc from the top, and this green plate here, this is the neural plate. Okay, and they've given you a set of cross sections as well, so you can follow along to see what's happening. So here you can see how the neural folds have started to elevate, and they're growing towards each other. In this third picture here, they've fused together in the midline, and like I said, the fusion is going to continue cranially, so towards the expanded end of the embryo, and is going to continue caudally towards the lower ends of the embryo, and then eventually you have a completely formed neural tube. So when the fusion is complete, the structure that is formed is called the neural tube, and you can basically think of it as looking like a lollipop, okay, because it has a narrow part caudally, so the stick of the lollipop. This will become the spinal cord. The cranial portion, the portion that's closer to the head, is dilated to form structures called the brain vesicles. So that's the sweet part of the lollipop. Okay? And the brain vesicles will end up becoming the brain. Okay? Now, something to keep in mind is a population of cells called the neural crest cells. Now, while the neural folds are changing, so while the whole process of elevation and fusion in the midline and all those things are happening, a mass of cells will detach from the lateral part of the neural plate. Okay, so if you remember with the initial picture with the 4 stages from A to D, even though the neural folds were elevating and fusing, there was still a little part of the neural plate left laterally. So a mass of cells will detach from that part of the neural plate, and they will migrate. Basically, they're going to start moving around the different parts of the developing embryonic disc. Now, depending on where they go, these neural crest cells become different things in the future. Okay, now remember the embryonic disc is flat. Okay, so there's a certain number or limited number of places they can go. Okay, so some of them are going to migrate dorsally, meaning they will go upwards towards the ectoderm. Okay, so they're basically going to go up and become closer or part of the ectoderm. The ones that go to the ectoderm are going to become something called the melanocytes of the skin. And these are the cells that are responsible for skin color. They're basically cells that release the pigment melanin. Okay? Now, the ones that migrate in the opposite direction, and they go ventrally, so this means downwards towards the mesoderm, they will become the neural cells that control the internal organs. Now, remember that in terms of control or the things that the nervous system does, you have two types of control. You can either have conscious control or unconscious control. Conscious control is the things that you decide that you want to do. You want to jump, you want to run, you want to walk, you want to sit down. Unconscious control is the control of the internal organs that makes sure that they function properly, but this is not something that you can control. So things like your heart beating faster, or your gastrointestinal system contracting more, or the breathing movements of your lungs and the expansion and the things that happen there. Okay, these are things that you can have some conscious control over your breathing, but for the most part, you do it unconsciously. So, even though we don't think about these things, they still need some form of nervous control. So, the cells that control our internal organs originally come from the neural crest cells, but the only difference is that they migrated in a different direction. They went towards the mesoderm. The last option, or the last group of cells, detach, and they actually detach from the cranial end, and so they basically migrate towards the head, and they become the craniofacial skeleton. So the bones of the skull, the bones of the face, originate from this population of cells. Okay, so let's just look at this as an animation for you guys to kind of get the feel of the things that I was just explaining. Okay, so this is a sagittal section. It's showing you all the different parts of the developing disc. Okay, this is again an aerial view. Okay, and the notochord has appeared and has induced formation of the neural plate. Okay, and so you can see the notochord kind of under the neural plate, and this is the part where it starts growing towards the primitive streak. It's expanding. Okay, and now the neural plate has increased in size. Okay. So you're going to look at this in a cross section just to make it easier to understand. Okay, these are all the different parts. You've got the neural folds, the neural groove. They're going to start fusing and growing towards each other. And then look at this population of cells, okay? So notice how this population of cells kind of detached from the side of the neural plate, and those were the neural crest cells. Okay, so let's just go back for a second. Sorry, my bad. Okay, so once they fused, this population of cells detached, and they formed the neural crest cells. So these are the cells that are going to start migrating and moving all over the place. Like I said, they can either go dorsally towards the ectoderm, so up here, or they can go ventrally down here towards the mesoderm, or they can detach from the cranial portion of the neural plate and end up giving the craniofacial skeleton. Okay, and we're just going to look at the formation again from a different viewpoint. Okay, so it's just showing you how the folds are going to end up fusing together in the midline. And you can see how part of it is expanded here, and see, it happened craniocaudally. Okay, and now you have the formation of the neural tube. Okay, and now the fusion is complete. These are the dilations that I mentioned at the cranial end, which are going to become the brain. And then obviously it's narrower down here, so this is going to become the spinal cord. Okay, so you can see here how a little part of the structure is starting to appear. Okay, so you can see here how this is clearly going to be the head. You can clearly see how this is obviously going to be the spinal cord. These structures here, we're going to talk about them in a minute. Okay, but again, this is something similar to the vertebral column, if you've seen one before. Okay, so now the embryo is starting to take a more clearer form. Okay, so we're going to stop here, okay, because I just wanted you guys to see the formation of the neural tube. Okay, so that concludes the development and changes happening in the neural tube. All right, now we're just going to continue with all the other different things that happen in the ectoderm. So we have structures called placodes, or if you want to call them placodes, I like saying placodes, it's better. It sounds more natural to my ears. So the placodes are ectodermal thickenings that appear in the head on either side, so bilaterally means on either side. And this happens by the time the neural tube completes its closure. Okay? There are two types. We've got something called the otic placode, and we've got the lens placode. And without going into too much detail, because you're going to get this way, way down in the future. Okay, but essentially the otic placodes will become the structures of the ear, and the lens placodes will end up giving me the lens of the eye. Okay, so for these two, I just need you to know that they are ectodermal thickenings. You need to know their names and what they become. All right, and then just a quick look here. Okay, this is where the lens placode is. So obviously it's the region of the future eye. And then over here you have another ectodermal thickening, which is going to become the future ear. Okay, so in summary, the changes that happen in the ectoderm are going to give the central and peripheral nervous systems, which we saw with formation of the neural tube. They give me parts of the eyes, the lens, and parts of the ears. Okay. And the ectoderm, even though we haven't gone into the specific details of how this happens, but the ectoderm also gives rise to the epidermis, which is the outer layer of the skin. Okay, so hopefully you know, or we'll find out at some point, that the skin is actually made of two layers, an outer layer called the epidermis and an inner layer called the dermis. So only the outer layer of the skin comes from the ectoderm. And if you want to remember the structures that originate from the ectoderm easily, just remember that any organ that maintains contact with the external environment has to originate from the ectoderm. So you feel things with your nervous system, you see with your eyes, you hear with your ears, you feel things with your skin. Okay, so anything that maintains contact with the external environment originates from the ectoderm. Okay, so hopefully the development of the ectoderm is clear. Now we're going to move on to the changes and development that happens in the mesoderm, which was the middle layer of the trilaminar disc. Okay, now it is initially arranged as a flat, loose layer on either side of the notochord. So it's just basically a haphazardly arranged mass of cells, just kind of sitting there on either side of the notochord. Now, by day 17, what happens is it starts to rearrange itself. So instead of becoming just this loose, random layer, it rearranges in a way that it forms a thickened plate near the midline. Okay? We refer to this as the para-axial mesoderm. Further arrangement, or this arrangement is in general, gives me the intermediate mesoderm right next to the paraxial mesoderm. And anything remaining at the sides is referred to as the lateral plate mesoderm. So basically, at the beginning, it's just a flat, loose layer of cells, and then they start to organize themselves so that they can be divided into 3 separate segments. Okay, so this is a cross section. Okay, like I said, anything in blue is the ectoderm, anything in yellow is the endoderm, anything in pink or red is the mesoderm. So in the middle here, we've got the notochord. Okay, and notice how you have a well-defined mass of mesoderm on either side of the notochord. That is called the paraaxial mesoderm. Right next to the paraaxial mesoderm, even though there's no specific line here that you can see, but right next to the paraaxial mesoderm, you have the intermediate mesoderm. And then at the sides here, where it kind of starts to flatten out, this is called the lateral mesoderm. Okay, with further development, so now we have the mesoderm divided into 3 parts. Now, with further development, we have the appearance of cavities within the lateral plate. Okay, so if you remember, if we go back to the picture, notice here how the lateral plate mesoderm is a single layer at this point. But once the cavities appear within the lateral plate mesoderm, it's going to be divided into 2 layers. Okay, I have an upper layer which is continuous with the mesoderm lining the amniotic cavity. Now remember, we had the extraembryonic mesoderm that formed last time. We divided it into 2 layers, one that lined the developing embryonic disc and one that lined the cytotrophoblast. So the lateral plate mesoderm, once it starts dividing into 2 layers, the upper layer is continuous with the extraembryonic mesoderm that lines the amniotic cavity, and we call this the somatic, or you can also call it the parietal mesoderm. Okay? The lower layer is continuous with the extraembryonic mesoderm that lines the yolk sac, and this is called the visceral mesoderm. Okay, now I know this is a whole bunch of new names. Okay, so let's look at just one more thing before we move on to a picture form. Okay, the cavity that appears in the lateral plate mesoderm to divide it into 2 is called the intraembryonic coelom. Okay, and this will eventually end up becoming the cavities of the chest and abdomen when the embryo folds. Okay, now I know that was a lot of information, so let's just look at it in video, and hopefully this will make it clearer for you. All right. Okay, so first of all, let's just get our bearings. Okay, we are looking at a sagittal section. Okay, so basically they've cut it from top to bottom, and you can see the length of the developing embryo. Okay, this is the trilaminar disc. You're looking at it from the side. Okay, anything in blue at the top here is the ectoderm. Anything in red is mesoderm, and anything in yellow is endoderm. On top here, you've got the amniotic cavity. This is the cranial end where the head is going to develop. This is the caudal end, which is the lower part of the body. And down here, you've got the yolk sac. All right. Now, I want you to notice how at this point, the embryonic disc is flat. Okay. Now, let's see what's going to happen. Okay, now this is a cross section, all right, and again, let's just identify some things. Okay, again, this is the ectoderm. The red part is the mesoderm, and notice how it is, well, I mean, you can't see the divisions here, but you've got the notochord, which is the green line, green circle here. You've got paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm here. And this is the endoderm. Now remember, this is the amnion, this is the yolk sac. Notice how Each one of these cavities is lined by what we call extraembryonic mesoderm. And if you follow the lateral plate mesoderm, it's actually continuous with this layer. Okay? So you've got this continuous with the amniotic extraembryonic mesoderm, and you've got this one continuous with the yolk sac mesoderm. Okay, now you see what's happening? The neural folds are rising, and at the same time, the sides of the amniotic cavity are actually kind of coming closer to the midline. Okay? All right. See the lateral plate mesoderm here, how it's divided into two layers? Okay. Now, notice how the ends of the amnion are going to come closer and closer until they kind of crush the yolk sac. And at the same time, I want you guys to follow this cavity here, okay? Remember, that's the intraembryonic coelom, all right, which was the cavity that divided the lateral plate mesoderm. Okay, see the intraembryonic coelom here? What's going to happen is eventually these two are going to close, and this is going to become the body cavity. And notice how now the lateral plate mesoderm is actually divided into two parts. You've got one here and one here. Okay, so just one last view so that you guys can kind of see it properly. Sorry, my bad. Okay, so notice how the intraembryonic coelom kind of got larger with this folding and this fusion. Okay, and this is now the body cavity. Okay, which means that this is the part where, you know, in the chest, the heart and the lungs are going to develop. In the abdomen, you've got the development of the GIT. Even you can see it here, it's called the embryonic gut tube. This is where the gastrointestinal system is starting to develop. So, like I said, the intraembryonic coelom will end up becoming the body cavity.